So now we'll discussing what are the different types of transport mechanism. As you already know, transport mechanism. What is transport mechanism? They are the ways which substances can be transferred from one place to another. Let's see what are the different types of transport mechanisms. First, we'll discuss about passive transport, and the first one is simple diffusion. In simple diffusion, solute in gases enters into the cell passively. What is passively? Passively means it travels from high concentration to low concentration. It can also be called along the concentration gradient. It is driven by the concentration gradient or the pressure gradient, since for a gas. And the rate of entry is directly proportional to the solubility of solute in hydrophobic core of the membrane. Suppose there is a molecule and its solubility in the membrane is very high as compared to this molecule, which is polar in nature. So it will be difficult to pass for it, but it will easily pass the lipophilic molecules, because lipophilic molecules find it easy to pass to the hydrophobic core of the membrane. And it also depends on the concentration gradient. Greater the concentration gradient, greater the mobility will become. Since it is a passive transport, it does not require energy, means ATP is not used, and it's a very slow process. The next uh, type of passive transport is facilitated diffusion. As it is written in its name, which is called facilitated, means the transport is helped or it is facilitated by something, and the something which helps or facilitates the transport is called carrier protein. In this, the movement of particles along the concentration gradient take place. Along the concentration gradients means from high concentration to low concentration. With the help of carrier protein or transport proteins, there's a special group of proteins which helps in transport. Since it's a carrier mediated process, what do you mean by a carrier mediated process? It means that a carrier is required for this type of transport. It follows saturation kinetics. What is saturation kinetics? Let me tell you what saturation kinetic is. Suppose this is a cell, and in this cell there is 3, 4, let's suppose there are 4 transport proteins which can promote facilitated diffusion, and we have 6 molecules. So at one time, how many molecules can pass? 1, 2, 3, and 4. So if I increase the amount of molecules, will they able to pass through the membrane? No, because the carrier protein is limited. So saturation takes place at a certain level. Therefore, it follows saturation kinetics. It's a passive downhill process since no energy is required. Downhill process. It follows inhibition kinetics and can operate bidirectionally in either direction. Example of this type of transport are glucose transport, galactose transport, leucine and other amino acid transport. As we know, glute, what is glute? Can anybody tell what glute is? Glute is transport of glucose by facilitated diffusion. Water or aquaporin, both are facilitated diffusion example. Let's understand the example of facilitated diffusion. Mechanism of facilitated diffusion. It occurs by a ping pong model. What is ping pong model? Let's see. So this is the structure of a ping pong model. This is the ping conformation and this is the pong conformation. Ping means narrow, pong means open, so that the solute can fit in this side. And when the solute fit in this configuration, it changes the overall structures and exposes the solute molecules on other side in pong formation. Let's see the step. Pong conformation of carrier protein is exposed to the solute side. So it is exposed to the higher concentration of molecule which is to be transported. When the binding of solute takes place, this solute binds to the Pong conformation. What happens is there is a conformational change in the carrier protein to ping state. So there is a change takes place. This Pong conformation is changed into ping, and this ping conformation changes to Pong. And when this changes into Pong, the solute is automatically exposed to the lower concentration side. And when this happens, the solute is exposed to the low concentration time, which resulted in release. There is a conformational change to the carrier protein back to the Pong form, and it again becomes this, and it goes on until the concentration is same on both the sides. So this type of accelerated diffusion occurs. Now let's talk about the active transport. Active, which actively requires energies. It requires ATP. It is also carrier mediated. It requires specialized integral proteins called transport proteins, or it can be transport pumps. It takes against the concentration gradient. What does that mean? Means it is an uphill process which requires energy, ATP, adenosine triphosphate. It follows inhibition and saturated kinetic, and it is a unidirectional process. So these are some examples which are transported through active transport. So active transport are of two type: primary active and secondary active. Primary active, in its name it suggests that transport of substance against its concentration gradient with the utilization of energy directly. Sodium potassium ATN, calcium ATPase, hydrogen pump, and here ATP is used indirectly. Glucose sodium co-transport, amino acid sodium co-transport. So these are the examples. What is symport? Symport is when two different substances are carried across the membrane in the same direction. Something like this. Example of this is glucose sodium and amino acid. Antiport, when two different are carried in opposite direction. Two different substances are carried across the membrane in opposite direction. Sodium calcium and sodium hydrogen. So we have understood that glucose sodium is an example of symport, and amino acid sodium is an example of symport. But sodium calcium and sodium hydrogen example of antiport. You can also remember that the antiport example, sodium is a positive ion, it is a cation, and calcium is also a cation. So when suppose the sodium is going into the membrane, so calcium have to come out to maintain the electrical neutrality. So somehow, even if the electrical neutrality is changing, so somehow it levels the does not change the electrical neutrality too much. Suppose Na plus is going in, and calcium is under, so too much positive charge will accumulate in this, and it can disturb the electrical neutrality. So if the ions are same, then it must be mostly antiport transport. So let's see how sodium potassium ATPase work. So sodium potassium ATPase, it is an integral part of the membrane first of all. So what happens in sodium potassium ATPase is it pumps out 3 Na plus outside and 2 K plus inside. 3 Na plus outside, 2 K plus inside. And it happens against the concentration gradients, mean is using energy provided by 1 ATP hydrolysis. It is the most common pump found in all the parts of the body, and it uses membrane bound ATPase as a carrier molecule. What is membrane bound ATPase? It is a carrier molecule which is used in sodium potassium ATPase, an enzyme that catalyzes the hydrolysis of ATP. So this ATPase, membrane bound ATPase, has 6 subunits, 3 alpha and 3 beta subunits, which is extended all throughout the membrane, and only alpha subunit is responsible in sodium potassium ATPase activity. So sodium potassium transport occurs through the alpha subunit, which has ATP enzymatic activity, which helps in ATP hydrolysis. ATP will be converted into ADP plus IP and then energy. And the next is binding site, intracellular for 3 Na plus and 1 ATP molecule, and extracellular for 2 K plus. So there are total of 3 subunits, 5 binding sites. 5 binding sites for 3 Na plus and 2 K plus. Subunits are 3, which is alpha 3 subunits, and responsible for sodium potassium activity. So this is the diagram. As you can see, this is the alpha subunit, this is the beta subunit. It has ATP binding site, phosphorylation site, binding site for 3 Na plus, and the opposite side are binding site for 2 K plus. As I've also mentioned the steps, binding of 3 Na plus and 1 ATP to carrier protein inside the cell transfer the high energy phosphate from ATP to aspartic acid residue of alpha subunit of ATPase, which what happens by that, phosphorylation occurs, which causes change in the shape of protein, resulting in 3 Na plus movement out of the cell. And what happens after that, 2 K plus bind to the external surface, and the aspartic acid bond get hydrolyzed, which occurs dephosphorylation, and causing second change in the protein shape resulting in 2k movement inside the cell. So this is the rough diagram. 3Na plus will move outside when phosphorylation occurs, and 2k plus will move inside when dephosphorylation occurs. As we know that Na plus are moving inside, no, K plus are moving inside and Na plus are moving outside, so Na plus concentration will be high outside and K plus will be low outside, and Na plus low inside and K plus will be high inside. So let's talk about some function of the sodium potassium pump. Sodium potassium pump works non-stop and is responsible for maintaining the high K plus and low concentration of Na plus in the cell. It is an electrogenic pump, coupling ratio 3:2. What is the coupling ratio? How it is coupling the ions? It is exporting 3Na plus outside and 2K plus inside. Therefore, coupling ratio is 3:2. It causes the movement of positive charge out of the cell. So if 3Na plus moving outside, 2K plus coming inside, so 3 minus 2, 1 positive charge is moving out of the cell. So net positive charge is moving out of the cell. It accounts for a large part of the BMR. Since it is a major energy using process, it requires ATP, adenosine triphosphate. Helps in regulation of normal cell volume and pressure, since cell volume also depends on ion concentration and all. Let's talk about inhibition of the pump. If the concentration of sodium and potassium ion are low, then the activity of sodium potassium ATPase is also low. If there is low temperature, if there is lack of oxygen, metabolic poisons like 2,4-dinitrotoluene, digoxin, ouabain, dopamine, these also inhibit the sodium potassium pump. Increased activity of sodium potassium pump takes place in presence of insulin, aldosterone, G-actin, and thyroid hormones. Digoxin is also used in the treatment of congestive heart failure. This is a one marker asked in many papers. So these are also the key points which you can remember. Next, the sodium potassium ATPase was an example of primary active transport. Let's understand secondary active transport. Secondary active transport example is sodium glucose transport and sodium and some other amino acid transport. So I'll explain this by giving the example of sodium glucose transport. In sodium glucose transport, as we know that sodium potassium ATPase transport the Na plus out of the cell and put K plus inside the cell. And this process takes place with the help of ATP creating sodium plus gradient, because we are creating more Na plus outside and low Na plus inside, so there is a high concentration of Na plus outside the cell. So this Na plus wants to come inside along with concentration gradient. So when this Na plus comes inside, what happens is glucose comes with it. Na plus moves along the concentration gradient into the cell, pulling glucose along with it against its concentration gradient. Hence, energy is utilized directly. See, 3Na plus moves outside, 2K plus moves inside, and now this Na plus wants to move inside along the concentration gradient. But when it's coming in, it pulls glucose along with it against the concentration gradient. Glucose is coming against, but sodium is coming towards the concentration gradient. So this is an example of secondary active transport, since ATP is not utilized directly as in case of sodium potassium ATPase. ATP is used indirectly. So this is sodium glucose importer. So glucose will come on and facilitate transport, it will reach the blood vessel. Some other points to remember: the size of the molecule, charge of the molecule, type of molecule. These are the things in which selectivity of membrane depends. Let's talk about ion channels. Ion channels, what are these? Ion passes through ion channel which open or close in response to a signal. And these signal can be two types. Either it can be voltage gated or ligand gated. Voltage gated means it opens due to changes in the membrane potential. Membrane potential means the membrane voltage. Calcium, sodium, potassium channel are example of voltage gated channels. And we have ligand gated channel. Binding of ligand to receptor site resulting in opening and closing of the channel. Example are acetylcholine receptors present in the postsynaptic membrane. Ion channels are important for nerve impulse transmission, synaptic transmission, and the secretion of biologically active substances from the cells. Endocytosis. Endocytosis is uptake of macromolecules into the cell, and uptake of low density like lipoproteins, polysaccharide proteins, and polynucleotides. Pinocytosis is uptake of fluid and fluid contents by the cell. It is also called cellular drinking. Phagocytosis, ingestion of larger particle like bacterial cell and tissue debris by macrophages. And exocytosis is release of macromolecule from cell to outside. It is exo, and ingestion is endo. Example are calcium dependent secretion possibly from secretion from the hormones. As we already know what is uniport, symport, and antiport is. Uniport is single molecule transport into a cell. Symport is two molecule in same direction, and antiport is two molecule in opposite direction. Example as is fructose in intestine, example of uniport. Sodium glucose transport in the intestine, example of symport. And chloride hydrogen, as we know, it occurs in RBCs in CO2 transport. Chloride hydrogen transport in RBCs. What are aquaporins? Aquaporins are basically water channels. It controls the water content of the cell. It is a family of membrane channel protein that serves as selective pores through which water crosses the plasma membrane of the cells. Facilitated diffusion example is Nephrogenix insipidus, which is due to impaired function of these aquaporins. That is one of the things. And channelopathies. What are channelopathies? These are a group of disorders that result from abnormalities in the protein resulting in the iron pores channel. Cystic fibrosis, periodic paralysis, and Little's syndrome are an example of these.